Good afternoon, everyone, and welcome to today's virtual information seminar on congenital muscular dystrophies. My name is Romla Kadia, and I'm the information coordinator here at Muscular Dystrophy UK, and I'll be chairing today's session along with my colleague John, who is our research manager. If you have any questions about the session content, you are welcome to get in touch with us, and we may refer to congenital muscular dystrophies as CMD throughout this seminar. We're so pleased that you could join us for what promises to be an informative and engaging session. CMD is an umbrella term for a group of rare inherited muscle wasting conditions that usually present at or near birth. And while these conditions share certain features, each type of CMD is different and experiences can vary. This afternoon, we have a packed agenda with a fantastic lineup of speakers ahead. So the seminar will be split into two parts. We'll begin with a focus on research, followed by a chance to ask your questions on that topic. In the second half, we'll move on to discuss everyday life with CMD, including a general discussion and plenty of time for questions. Throughout the seminar today, you're welcome to submit questions using the Q&A function that will be at the bottom of your screen, and these will come through to us and we'll answer as many as we can, including some questions that have been submitted in advance. Unfortunately, we won't be able to invite people to speak directly, but we will try to make sure questions are brought into the discussion during the seminar. If we're unable to respond to any questions during the session, we'll aim to follow up afterwards and share additional information on our website as needed. While we'll do our best to cover as much as possible, some specific or personal questions may be better addressed by your clinical team or neuromuscular specialist. If you do not have access to a specialist, please speak to your GP. This session is intended to provide general information and does not replace individual medical advice. So just a reminder that MDUK's helpline is available to support anyone affected by muscle wasting conditions. Whether you have questions, need advice, or are looking for support, we are here to help. You can reach us on 0800 652 6352 or at info@musculardystrophyuk.org. Today's seminar is being recorded and the video will be made available on our website and YouTube channel in the coming days. Now I will pass over to John, who will be chairing the first part of today's session, and John will introduce our first two speakers.
Thanks, Romla. Welcome everyone to this section of today's webinar, which is devoted to research into congenital muscular dystrophies. We have two guest speakers today, the first of which is Francesco Saverio Tedesco. Saverio is an academic and clinical researcher with an interest in muscle regeneration and pediatric neuromuscular diseases. He's been at University College London since 2012, where he's now a clinical professor of neuromuscular biology and neuromuscular medicine. He's also an honorary consultant in pediatric neurology at Great Ormond Street Hospital, and since 2020 he's been a group leader at the Francis Crick Institute. So welcome, Saverio. Thank you for agreeing to talk today. When you're ready, please go ahead with your talk.
Thank you, John. Thank you very much for the kind introduction. Let me just make sure that I'm sharing my screen properly. Hopefully you can see my screen.
We can.
Great, thank you. As I said, thanks for the invitation and for giving me the opportunity to provide a brief overview of some of the progress and the updates on current research in this broad field, the broad field of congenital muscular dystrophies. As I mentioned here in my slide, I will be mostly focusing on a specific form of muscular dystrophy as an example, almost as a case study, the one related to the gene LMNA. And I also take this opportunity to apologize for those of you who attended already the webinar last year and might have seen some of the slides already, in case they are a bit of a repetition, but hopefully that will be interesting for the majority of the attendees. Some disclosures. I mean, I have nothing really relevant in terms of conflict of interest for CMDs, but I work with some companies on some of the platforms and the the research you will see in this presentation from our lab. Particularly for our work, we mostly study and model muscle development and the way muscle degenerates in diseases and regenerates, to use those that information really to develop treatments for severe neuromuscular diseases of childhood. And we work at the interface, at the crossroads between these three large areas: the stem cell biology, disease modeling, and gene therapy. Now, a few words about muscle. I assume that we have a bit of a broad audience. There might be people more experienced and expert in this, and others who are new to the topic. So it is the most abundant human tissue and the main output of our brain. And this cartoon here shows you a bit of an overview of the structure of some muscle fibers. This is an example of one muscle fiber, in which you can already spot that there is something quite interesting, which is a lot of little dots here, which has this nuclei where the DNA is contained, where the genes, they have the instructions to make the bits of our body are contained. And we have several of those in each single muscle fiber, which is a quite characteristic thing for muscle. And we have connective tissue around, sort of a matrix wrapping up the fibers, and they are in close contact with blood vessels and the nerves, which bring the stimulus to tell the muscle which are the function that it's supposed to be doing. And those functions are multiple. You see here movement, supporting soft tissues, maintaining temperature, storing nutrients, and so on and so forth. And this is another view of how muscle is organized, and this will be relevant for what I'm going to tell you in a minute. You see an example here again of a muscle fiber next to a few other muscle fibers over here, all wrapped around this whitish layers of matrix, which is the connective tissue I told you before. And you see these little guys here over the muscle fiber underneath the layer of connective tissue, which are the satellite cells, which essentially are the muscle stem cells responsible for regenerating muscles generally. Now, why is this relevant for congenital muscular dystrophies? So first of all, what are congenital muscular dystrophies? Well, we heard already they are inherited conditions primarily affecting muscle and characterized by low muscle tone and weakness before, roughly speaking, before walking, if we want to really to simplify that, in the first 1 or 2 years of life. And often also with delay or arrest in some of the motor milestones that the children have to achieve. Now, when we then look at the muscle, how the muscle looks like, we say that the muscle is dystrophic, and that will be clear in the next slide. And a key thing that I will be stressing a few times during my talk is that, and we heard already some of it earlier, is that there are several different types of congenital muscular dystrophies, each one of them caused by a gene, changes, I should say, in genes, which are responsible to produce a number of different parts of our muscle fibers. You can already see in this cartoon here from a review from Alberto Zamboni and Francesco Montoni, you see here that these are in the white boxes some of the key genes that are causing the majority of the congenital muscular dystrophies. And you can already appreciate that they are scattered pretty much everywhere in the muscle fiber, from the nuclear compartment, the nuclear envelope where the DNA is stored, up to the outside of the muscle fiber where the collagens are. And this is going to be relevant as well for things I'm going to tell you in a minute. Unfortunately, the common aspect of all these different forms is that quite the majority of them are severe, and invariably all of them are to date are incurable. So we don't have really a cure for any of them. So it's really important to understand how they develop so that we can also develop therapies. Now, I told you about the muscle biopsy, which is, we say, dystrophic. And what does this mean? Well, this is how a healthy looking muscle look like if you do a biopsy, if you sample a bit of it and you want to, and you look at it under the microscope. Now we're looking at those fibers, not like longitudinal, like top to bottom as we've seen before, but we're looking at them from the top. right? Imagine that, we're slicing a muscle like the bicep here, and we are looking at it from the top or from the bottom. These are the muscle fibers that we see that pretty much all of them look similar. Now, this is what a dystrophic muscle will eventually look like. And you can already appreciate the big difference. You see that the shape of the fibers is very different. We have lots of little dots here in purple, which are inflammation, cell of the inflammation that are scattered around the muscle fibers that are responsible to clear up the fibers which are dying and help regenerating them. But this process sometimes doesn't work properly, and we have the swapping of the muscle tissue sometimes with scar tissue or with fat tissue. And if you want to imagine essentially how they should kind of look like in reality with an analogy, imagine those fibers as ropes that have the nice little intact core and the outer bit which is shielding them from damage. And this is how the fibers, the ropes, will look in a healthy condition. In a dystrophic muscle, the ropes, the fibers, will have different sizes. Some of them will be very thin, so very prone to rupture. Some of them will have the outer layer, the sheet of the ropes, which is very fragile. And indeed, when the muscle twitches, they will just break down like this. So this is just to give you a rough idea of what will happen at the level of the tissue. Now, I told you that I will be focusing mostly on one example because giving you insights on all of them is really tough, besides not being an expert in all of them myself. I'll be focusing mostly on the congenital muscular dystrophy caused by changes in this gene called LMNA. My lab does a lot of work also in another congenital muscular dystrophy, which has a very different mechanism, but to some extent similar problems to this one, caused by mutations, by gene changes in the collagen 6 gene. And we're talking now in a problem which lies outside of the fiber rather than inside like this one. But there are, as I said, several commonalities between them. And we can discuss later on some other specific aspects of other forms if you wish. But I think the LMNA one gives a very nice case study because of the complexity and the involvement of also other tissues that we have in this condition. So a few words about this bit that doesn't work properly when we have a gene change in LMNA. This bit is the nucleus, where the genetic information, where the instructions to make the proteins of our body are stored. And the nuclear envelope is this sort of layer which wraps around the DNA. And particularly in our specific case, we have this orange bit, which is the nuclear lamina. So it's sort of an additional layer underneath this envelope, which helps, again, regulating how the genes are turned on and off, and also gives stability to the cell nucleus, almost like a nice bookshelf, nice and stable bookshelf for all the books which are the genes inside this compartment. So when that compartment doesn't work properly, we have a group of more than 15 conditions called laminopathies, which belong to essentially 5 main groups: cardiomyopathies or heart problems, muscular dystrophies, which is what we're going to be discussing today, nerve problems or neuropathies, or problems of the adipose like the fat tissue, and premature aging problems like the progeria, which you might have heard of. In the case of the muscle ones, the one of interest for today, we have problems, as we said, which are both affecting the movement muscles, or the skeletal muscles, and the heart muscles, with a cardiomyopathy. Now, and we tend to call them collectively, like altogether, as striated muscle laminopathies, because striated muscle are essentially, striated muscles are the heart muscle and the skeletal muscle. But for simplicity, we can even just limit today's discussion to call them muscle laminopathies. So these are some of the important initial papers that were published to describe those conditions. And as I said, we have problems with the heart, with a condition called dilated cardiomyopathy, which is almost invariably common in problems with this gene. And when it comes to skeletal muscle, we have muscular dystrophies. We have several different types of muscular dystrophies, actually 3 main types. One is called Emery-Dreifuss muscular dystrophy, characterized by muscle wasting, weakness, and contractures, and heart problems. Another one is called limb-girdle muscular dystrophy 1B, which is similar to this one, but similar to the Emery-Dreyfus one, but with less contractures. And then we have the LCMD, which stands for LMNA-related congenital muscular dystrophy. So this is the focus of today's discussion in my presentation. And this is the most severe muscle laminopathy. It has an onset, as I said, usually around, before the second year of age. And we have severe wasting of the muscles and weakness. And again, you can appreciate that you don't have to be a pediatric neurologist, that in some severe cases of LCMD, we can see very severe presentations like in this case here. We have problem with the spine and often and almost invariably heart problems, breathing problems, and also problems with feeding. Now, what's happening in this particular condition in terms of research? Well, there are many different types of research activities going on. I'm going to start from clinical research, and I will go down to preclinical and more lab-based work, which is what I'm also doing quite a lot here at the CRIC and at UCL. So in terms of clinical research, there is active research ongoing in natural history studies, those who understand really when are the problems developing and what are the consequences of those over time. And you see an example here on the right of an important study published a few years ago, led by Giselle Bon and the group Dystomyology in Paris, where they analyzed a large international group of individuals with LCMD to define really where are the complications usually arising and what are the associations of those complications with specific gene changes in the LMNA gene. There is active research ongoing on the so-called disease modifiers. So what is causing individual A to have milder symptoms than individual B, even though they share the same gene change, for example. And there is active research ongoing in outcome measures and biomarkers. So to understand really what can be monitored both in the lab and in clinical studies in the hospitals to understand the disease progression, but also the potential amelioration if we then start a treatment. And there is also active research in terms of those treatments that I just mentioned. One of those is not so surprising, which is steroids. And most of you would have heard about the use of corticosteroids of these drugs in Duchenne muscular dystrophy. There is a lot of interest in some congenital muscular dystrophies as well to try steroids, and they have been tried not systematically, but they have been tried in this particular CMD, in LCMD. And there is suggestions that there might be some specific group of children who can respond if treated very early. And particularly this is supported by the fact that there is a lot of inflammation ongoing and detected in this particular condition. But more studies are needed really to determine who and when and for how long could be treated with the steroids to sort of slow down the disease progression rather than having a cure in this case, but still very important. There have been trials in other aspects of LCMD, particularly the cardiac aspect, with a drug in this particular case called a P38 inhibitor. This was more for the cardiac problems, the dilated cardiomyopathy. This went up to phase 3 trials, so quite advanced clinical experimentation, unfortunately with little success and little improvement. Again, this is looking at the heart and not at the muscle problems, but obviously they're both equally important in this particular CMD. And then there is a lot of research happening in the labs, and I call them here as basic and preclinical research. There are lots of different animal models that have been developed over the last couple of decades for this condition, from worms actually, with showing similar problems, and you will be surprised how good they could be, up to flies, and then mouse models, some of them with specific gene changes that are also observed in the patients, others with some limitations in the sense that the mechanism causing the disease in human is very different from the mechanism that's been observed in mice. And also there are obviously vital, important patient-derived material like the biopsy material that we collect for diagnostic purposes. So when we have to make a diagnosis to help understanding if there is a defect in the way the muscle is made in this patient, and so on and so forth. But these models as well have some limitations that will be clearer in a minute, because we don't have an unlimited supply of those. Obviously, we can't go back over and over again and take several samples from an infant with a congenital muscular dystrophy. This will be unethical, will be invasive, and sometimes quite complicated. And there is also basic and preclinical research, not only on the mechanisms of the condition, but also on potential therapies. And there is active research in genetic therapies, and this will be clearer later on as well. But there are a number of different strategies that are used for that. Again, this is not very different from what's happening for other congenital muscular dystrophies caused by different genes. But here we have so-called RNA-based therapies, antisense oligonucleotide, you might have heard some of these names, and gene therapies done using specific engineered viruses that are made harmless so that they can be delivered to patients. And one such example, which is very promising, is again focusing on the heart problem in this particular CMD, which is it's been developed by a company which I believe is called Nuevocor, and they're about I think they're already in a large animal experimentation, if I'm not mistaken, but again, it's targeting the heart problem. There are gene editing strategies, so there are specific molecules or viral vectors or harmless viruses that are delivered to do genomic surgery, so to cut and paste, to fix the gene change on those nuclei that we saw before in the muscle fibers. And obviously there is a lot of different molecules, and there are different drugs that, for example, were used in a condition like the steroids, for example, that have been tried in a different condition, like in the case of LCMD here. And then there are a number of new emerging technologies, such as induced pluripotent stem cells, organoids, and beyond, that are used in a few labs, including mine. And we actually do quite a lot of work specifically on those new technologies to understand both the problems behind the onset of the condition and the potential to try them for developing new strategies. And let me tell you a bit about that. So we are building up in my lab, in particular, models which are specific for the individuals with those conditions, that are scalable, so we can make them different size at different time, and we can also analyze the development of a condition from a single cell, like a single muscle stem cells, up to a full muscle, and they are good enough for us to understand the mechanism behind the onset of those conditions. So we do that by making muscle from a specific type of muscle stem cells in the lab, in a dish, which is called human-induced pluripotent stem cells. The name is very complicated, but the concept is relatively simple. We can take, for example, a skin cell, take that skin cell from a little biopsy, or even a blood sample, actually. Take those cells, put them in the dish in the lab, give them a specific bunch of factors, and turn those cells into, let's call them superpotent stem cells, if you wish, which we call them pluripotent stem cells, which with the right cocktail, the right instructions, can be turned into any stem cell or any cell type of your body. This is a discovery that was awarded the Nobel Prize in 2012 to Shinya Yamanaka, and essentially with the right recipe you can go from a stem cell now to a full muscle fiber. And this is quite useful because it gives us an unlimited supply of patient-specific muscle-making cells that we can use in the lab to study the onset of the condition, why is that caused by a specific gene, and the response to specific therapies. And this is just an example of some of our early work in 2012, where we were the first to show that this could be done in muscular dystrophy, that you can take a cell, turn it into a pluripotent cell, and make muscle out of those that can also be genetically corrected. And then we actually applied this technology for the first time as a first target disease, specifically to LCMD. And we wanted to first see whether the abnormal shape of the nuclei, which was described in some of the biopsies in those patients, and you see an example here, could be seen also in the dish, in the lab, in those cells. And these are three variants from three different individuals with LCMD. And you see that actually this is from our stem cells, and by differentiating them into muscle, we will see similarly abnormally, atypically shaped nuclei of those cells, telling us that we were in the right direction. Now, these two graphs are quite complex here, but the take-home message is relatively simple again. So this abnormally shaped nuclei can be quantified, so we can tell how many we have. And we were expecting to see more going from myoblasts, which are, if you wish, sort of muscle stem cells, to myotubes, which are similar to muscle fibers. We were expecting to see more of this atypical nuclei, because that's where they are normally in the muscle fibers. And we saw less, actually. We saw a reduction in those. And we were quite puzzled by seeing that, and we wondered whether this was caused by the limitations in the way we were growing those cells in the lab. We're growing those cells as a monolayer, as a thin sheet of muscle cells in a dish, whereas in real life, the muscle is a highly three-dimensional tissue. It contracts, it absorbs shocks, and so on and so forth. So we wanted to have a better way to mimic that in the lab, a better way to really recapitulate the complexity of the tissue that I showed you before. And you see another example here with all the interactions with the other cell types in the culture dish. And so to do that, we pioneered really the development of this so-called mini muscles, really, or you might have heard them in other scenarios called as muscle organoids, from iPS cells, from patients with muscle conditions. We were quite good to go from one of those stem cells that I told you before, from skin fibroblasts, for example, to a myofiber or a myotube, the way we call them in a dish. But then we had to spend some time to learn to do the same thing also for other cells, like to neural cells, to blood vessel cells, so that we can combine them all together with special materials called biomaterials and make these little constructs here, which are about 1 centimeter long, which we call engineer muscle, or for simplicity, we can refer to them as mini muscles today. And this is one of our papers, actually the initial paper now, a few years ago in 2018, describing this approach. And this is more recent examples that I'm going to be telling you also some of the work in this paper today. If you want to have an idea about this mini muscles look like, now we're looking at, hopefully the video is working, but you will see the outer layer of the muscle fibers. In the central part of one of those mini muscles, you see titin, which is a protein very important to give contractility to the cells, and dystrophin, which I'm sure you've heard of, which is the protein that is not working properly in Duchenne muscular dystrophy. Just to give an example that in a normal healthy mini muscle, they are properly present in those mini muscles. These are muscles, and one of the advantages that we can do in this special way of culturing them is to make them contract. And now we can go much more beyond the simple analysis that we can do in a sheet of cells that if they contract, they will just detach and float around. Here now we can stimulate them, we can make them contract, we can make them twitch, and we can analyze if they are weaker or stronger, for example, before or after a specific treatment. And I told you also that we can grow them together with their friends, essentially. We can have the muscle fibers here. You see an example in gray, together with the motor neurons from the very same patient, in green, next to the blood vessel cells, which are yellow and red in this particular example. And back to LCMD now. So we took this technology and we took those iPS cells, those stem cells that we had from 3 LCMD individuals, and we look at the shape of the nuclei even more closely to see whether it is now more similar to what we would have expected in a normal muscle biopsy. And this is a healthy donor, and now you see the nuclei in red and the muscle fibers in green, and nothing particularly exciting. The nuclei have a relatively normal shape. But now when we look at the same in an LCMD individual, you see that they are very elongated here, very stretched. And that was quite a surprise for us. And this particular atypical shape was very rarely described before, and we did a lot of studies on that, and so that was also very much going hand in hand with the severity of the gene change. The most severe gene change will show the highest number of atypically shaped nuclei. And this was also then observed by other labs, and we're using it now as a readout, as an outcome measure to test for potential therapies. And as an example of those therapies, this is actual work that's been funded over the years and by Muscular Dystrophy UK, and I'm extremely grateful for the support, actually. And this is a pipeline that Daniel Moore, a talented PhD student, now postdoc in the lab, has developed. So we go from sampling the cells in specific LCMD individuals with mutations, with gene changes that we think are important to study, and we can look at modeling the condition, but also modeling the therapy in the same system, in using the iPS cells and using the mini muscles. And we can see whether that particular treatment, for example, improves the outcome in a specific gene change. And Dan has been trying this approach in two different ways. He's been looking at two different ways of doing so-called CRISPR editing, so the genomic surgery that I told you before, the copying and pasting and the fixing by specifically erasing and rewriting changes in the DNA so that we can now correct the misspelling that was there in the first place. And this is work that we posted on bioRxiv, which is a server in which you can download and look at the work before it gets published if you wish. This was there for the last 10 days or so very new stuff. And Dan has been essentially able to show, and this is a bit of a complicated slide, but I'll try to simplify it, that with our systems, you can determine if, for example, a specific type of correction with this editing approach, with the CRISPR approach, which was another a Nobel Prize awarded to two important colleagues over the last few years. And Dan was able to show that only when you do the full correction, so this, what you see as E wild type, so the look, you know, say it as the complete correction of the gene change, you see an amelioration comparable to the healthy control, to the healthy condition of those atypical nuclear changes. And you don't see the same level of correction when you do an approach which is called exon skipping, which is similar to what some extent has been done in Duchenne muscular dystrophy, where you get a shorter but still functional protein. Here we are seeing that we really need to correct completely these laminase proteins. So we really need to fix the scaffold of the nucleus completely to see an amelioration of those the shape of those nuclei, which we think correlates also with other parameters of the condition, including the force, and this is work in progress. And then before I finish, in the last minute or two, I just want to show you what we're doing now to go beyond just modeling the condition and beyond just trying these therapeutics as I showed you before, but try also to develop new treatments that can go into patients in a relatively short amount of time rather than in decades as we've been observing so far. Now, I'm sure that a lot of you are familiar with gene therapies, and particularly for the recent gene therapies in spinal muscular atrophies and in Duchenne muscular dystrophy. And what we're trying to do essentially is to use the systems that you've seen today, those mini muscles, to understand in a predictive way, so before they go into a clinical trial, which one of those vectors, which one of those gene therapies could be safer than others. And if, for example, in a specific condition, one type of vector, one type of gene therapy could be more successful than another. And this is very important because we've seen lots of successes in spinal muscular atrophy, for example, in SMA, but a lot of problems, including fatalities times, and you'll see this later on when it came to other muscular dystrophies or other neuromuscular disorders in general. So we want to try to avoid this and try to have safer and more efficacious gene therapy vectors that can be developed in humans in the first case. When I say in humans, I mean in human model, rather than having to go through a for example, an animal model and then find out that when it comes to humans, it doesn't work as well as it was working in the mice. And this complex cartoon here is to tell you that those modified viruses that are normally used in gene therapies, these AAV, adeno-associated viral vectors, are very many. You see here how many we have available. And all of them target different cell types with different efficiency. And knowing which one is going to work best in muscle or in other cell types which are important for muscle function is not that easy. Normally we do that in animals, but we don't tend to know that in humans very well. And indeed, this is sometimes what happens as a consequence of that. I told you about the great successes of muscle gene therapy, but our field in recent years has also witnessed a lot of side effects, severe side effects, some of them fatal, unfortunately, and those side effects are often side effects of, on the liver, on the heart, or on the immune system. And we want to be able to predict those side effects before it's too late and tweak the gene therapy so that they can be more efficacious in muscle and going less to the liver, for example. And to do that, I had the pleasure to put together this nice group of people in a consortium which I lead called MAGIC, which is focusing exactly on that, on trying to develop better predictive models of a number of different muscle disorders so that we can use them to develop next generation gene therapies that hopefully will give us the possibility to go faster into clinical studies. And I'm very proud to have also Muscular Dystrophy UK as part of this team. And just to give you a very simple example to give you a flavor really of what we're doing, this is an example of a viral vector of an AAV used for gene therapies routinely, AAV9 for spinal muscular atrophy, for example. You can imagine it that way, given in different doses to our engineered muscles, and you see that we have this fluorescent signal, which you see here in white as a readout, which is beautifully correlating with the dose that we're giving to this muscle over time. And this is something we can monitor, we can measure, we can correlate with a number of other measures at the moment, like force and so on and so forth. And stay tuned and check out our MAGIC Consortium website for regular updates on our progress on this in this area. So hopefully I convinced you on this last part today that also working in the lab, not only in late clinical experimentation, could be useful. And although all models sometimes are told to be wrong, we believe that some of them are actually useful, and hopefully that will be the case in CMD research in the upcoming years. So thank you again for your attention. This is our fantastic team. I acknowledge already some of the people doing the work. You've seen some of the work from Daniel Moore and Sumitava Dastidar in particular today, and some work by Valentina Lionello. And Kati Jiang is also leading on our collagen 6 work, which I didn't have the time to show you today. And these are our amazing collaborators and funders. And thanks again for your attention.
Thank you, Saverio. Thank you so much for such an interesting talk. There are one or two questions in the chat here. Bill, if I could ask you, I've just got a message to say he's figured out the answer. Okay, that's fantastic. There is a question about treatments, which I think maybe we'll come back to at the end after Anna has talked. Somebody's asking for a clarification, and they're saying for LAMA2, is the protein part of the extracellular matrix?
Yeah, I mean, LAMA2 is actually another very important gene-causing CMDs, and has an important role in the extracellular matrix, indeed. And to some extent, actually, he has a lot of similarities in the way we build up models for collagen 6, which is another protein that I mentioned before, very much involved into the stability of the extracellular matrix, and also in the in muscle regeneration per se. If you want, and if that could be of help, I can share again the slide with the scheme of where LAMA2 normally lives, but I something is happening on my screen. I've lost it. Oh, no, now I found it again. So you should be able to see my screen in a minute again. And we just mentioned Llama 2 before, and you see it here. Hopefully it works. You can see it on the screen. And you see it's quite important to link the extracellular matrix or the let's put it like the milieu, the matrix in that wraps around the muscle fibers, connects it to the actual muscle fiber. As you can see here, this is the inside of the muscle fiber. This is the outside of the muscle fiber. And this is the meshwork that we have outside of the muscle fiber, which connects it to the inside. And the two things have to work closely and together so that the muscle can properly functions. And what is a surprise for us and for many other people in the field, and this is becoming also very relevant for LAMA2, that although some of these genes and proteins like LAMA2, collagen 6 in particular, is not produced by the muscle fiber, but is actually produced, for example, in the case of collagen 6, by the muscle fibroblasts, the problem then is on the muscle fibers anyway. So you might have heard about this mechanism by technical people referred as non-cell autonomous. So even though it's a fibroblast, so it's a muscle matrix producing cell that has a genetic defect, eventually what doesn't work properly is the muscle fiber. And this is another area of active work that we're doing in the lab to understand what we could do to simplify, to understand the mechanism, but also to target the right cell types. And this is also a lot of work that is being done by Francesco Montoni and the rest of the team at ICH as well to try to target in particular those cells where the gene is causing the problem in the first case.
Excellent. Thank you for that answer. I think in the interests of time, I'm going to move us on. Thank you so much, Saverio. Please do stick.
Apologies for going a bit over time.
It's absolutely fine.
Sorry, Anna.
So I'm delighted to introduce Dr. Anna Sarkozy as our second speaker. Anna is an honorary senior lecturer and a neuromuscular consultant working at the Dubrovitz Neuromuscular Centre in Great Ormond Street Hospital. She's a major role in running the highly specialised service, which acts as a diagnostic and advisory service for congenital muscular dystrophies and for myopathies. She runs regular clinics for patients with rare neuromuscular conditions. Her academic interests are in the discovery of new genes that are important in congenital muscular dystrophies and myopathies, but she also has an interest in understanding the nature of rare conditions, to understand the natural history, of rare conditions. So thank you, Anna, for agreeing to talk. Please take it away when you're ready.
Thank you very much for this kind of introduction, John. And I, as you already mentioned, I'm going to talk about trial readiness, in particular natural histories. Saverio already gave a very detailed introduction, so I'm not going to repeat much in general on congenital muscular dystrophy. I will focus in particular what are the key steps that we really need to develop trials and therapies for rare muscular dystrophies, what are natural history studies, and use the example of LAMA2 gene-related muscular dystrophies to give you an overview of what we know and how we approach this issue for these rare conditions, and the importance of development of new diagnostic and treatment standards of care, and the role of registries. So just as an overview, congenital muscular dystrophies are early onset conditions, and as you already heard, are characterized by weakness, often presenting from the first months of life. Congenital muscular dystrophies are not the only one that can present with this problem at birth. There are many other conditions like those listed here. Congenital muscular dystrophies usually present with progressive weakness, very high CK, and with the dystrophic muscle biopsy that you heard already. And this is just a list of all the different types that are the most common types and how they're usually classified, classically also based on their muscle findings. as you heard, these are all rare and there is no treatment. So really the hope that we all have is that we are able to develop some therapies for this condition. Again, I'm going to put this slide back just to highlight again the high number of genes that are associated with these conditions. There are 47 known genes so far, but they are not all as common. You know, some are very rare and some are more common. And in the last few years, we looked into what are the frequencies of the various forms of congenital muscular dystrophies in the UK in particular. And this chart gives you an idea about the frequencies of all the congenital myopathies and muscular dystrophies. And this chart here on the left gives you an idea of only the congenital muscular dystrophies. And as you can see already, the three categories are jump out as the most frequent. So in particular, those related to collagen 6 genes, and these are very common. And then both in total among all early onset muscle conditions, but in particular among congenital muscular dystrophies, and then alpha dystroglycanopathies that include a large number of genes, up to 17, 18 genes now associated with this condition. But when we look at single genes, so one only gene causing a type of congenital muscular dystrophy, LAMA2 is the most common one, and this represents 15% of all CMDs. And then you have LMNA gene-related muscular dystrophies that you just heard about. So many, many genes, some more common, and definitely more prevalent among congenital muscular dystrophies. Now, there is no cure, but there is a lot we know about care. And this is very important, that we don't forget that while we wait for a cure, we really have to focus on developing and providing for patients supportive management and care with a multidisciplinary approach. And this is very important. And there has been quite a lot done in the last years. And I will come back later on this topic about the importance of management and international standards of care for children with muscular dystrophies. And a right management really starts with correct diagnosis that wherever possible should be genetic, based on genetic findings. So a correct genetic diagnosis should be really achieved for everybody wherever possible. And then MDT approach should include physiotherapy, respiratory, cardiac care, but also orthopedic and other aspects of MDT care. And of course, genetic counseling as well for the family. But of course, we hope for cures, and for that, clinical trials are important. And you already heard about development of new therapies for LMNA gene-related muscular dystrophies. So, but what are the key steps to develop clinical trials? And really, there are a few important steps that we all need to have for each of these conditions. And I will just touch base on some of these points. And explain why these are important to develop trials and be trial ready. So first, you really need to understand really well the natural history of this condition. And I will come back on a bit more in detail in the next slides. And in the meantime, you also need to make sure that children and adults with this condition receive optimal care. And this is why international standards of care are important. Why that is important, because clearly if you have treatments for patients with this condition, those who already have a good MDT care might respond differently to therapies compared to those who maybe are not availing of such opportunities. And we have to identify measures to identify response to therapies and markers that tell us how a condition progresses with or without therapies. And this may be different depending on the condition that you have. So really, we need disease-specific outcome measures. And some of these measures could be physiotherapy measures, like how fast you are to get up from floor, or how fast you are to walk, or for example, joint range. And then you have, or for example, respiratory abilities, how strong your lungs are to blow in a tube and so on. And also you have to make sure that these measures are able to capture change over time. So if I, for example, measure the ability to blow and your capacity to breathe over time, does this change over time, or does it stay stable? And if it changes over time, do you see a change in 6 months or in 12 months? Because clearly the trials that you develop may have to be at least long enough to show a difference over the time of the study. And then other biomarkers that have been being developed are, for example, application of imaging like muscle MRIs. And finally, another important step to develop therapies is to know the numbers of patients that have certain conditions. So I'll give you an idea of frequencies. We know that some conditions are rare, but what are the numbers? So if I want to develop a therapy, how many patients with a specific genetic defect or maybe with a specific gene variant could be able to avail of that therapy? And therefore development of registries and databases are very important to give us these answers and help us to become more trial ready. So each of these steps are really important. I would really like to start with natural history. What are natural histories? So we heard about this already, is really the quantitative understanding of what is the natural progression of the condition over time of a rare condition. And this is important to assess the efficacy of intervention, but also guide our counseling and anticipation of care. So if I know, for example, that a person with a condition X or Y will develop respiratory problems when they are 15 or 20, then I can start measuring the respiratory function and also develop intervention. So if I know that they might be at risk of developing some problems, I can start intervening early, maybe proactively to prevent. And this natural history and knowledge of natural history can also inform future clinical studies because it can tell me what are the outcome measures or the biomarkers that can be applied for that specific condition. So for example, if I know that a lung function ability does not change over time in a certain condition, maybe that is not a good outcome measure if my treatment cannot improve that function, and so on. So really, it's helpful to design clinical trials. So depending on what you need a natural history study for, there might be different type of natural history studies, so where you learn and study more the natural history of a condition. You can do cross-sectional studies when you take a group of patients with the same condition and you observe them, and then you give an information about how the patients present at that time. Or longitudinal, so you want to look over time how certain functions and abilities change and vary in the lifetime of a person. Some studies can be retrospective, so you go backwards. So you look at, for example, patients who are today with a condition, and you learn and you look in their clinical records and to see how certain functions were in the past. Or prospective, so you select a group of children and you see them, or adults, and you see them regularly over time, and you measure certain functions and abilities. And different studies, so you can do chart review, so you look at the clinical records. You can collect information through registries where patients upload information for themselves or their family members into the registries, or proper clinical studies like protocol-driven natural history studies. So these are the three main categories of type of studies that you can have. So going from chart reviews to protocol-driven natural history studies, the quality of data increases. So if the chart reviews have the lower quality data because you just get the data that has been collected, while in protocol-driven studies, you decide what are the information that you want to collect. But clearly, these also take a lot of personnel and time and resources. So, but it is very important that you know this, because really, if you don't know what is the disease course in a patient who has a treatment compared to patients who without a treatment, you wouldn't know if your therapy is working. And looking, for example, here, give an example of a motor function. So this is a person who has doesn't have a muscle condition. This is an idea of how the motor function progresses over time with an improvement. And then you have a person who has a condition where the motor function decreases. And then you have maybe a therapy that maybe start at a certain point and you can see there is a stabilization. But sometimes if you have maybe a cure, you would expect that the person's motor function would be similar to a person who has no treatment. So really knowing what is the natural history is important. And also knowing what is the natural history depending on the specific genotype, so the specific genetic defect, is even more important. So if you look at these three lines, so you have gene 1 in blue, the gene A in red, the gene 1 with the one type of genetic change, the same gene with another genetic change in red, and then you have in green another gene, but is similar to the progression over time to gene 1 in the red line. So this seems to be, the two variants here seem to have very different course, but sometimes the course of the disease can be very similar in early age. So it's very important also to understand what is the course of the disease at different phases of life. So it's important to understand early natural history, also to help to distinguish not treatment effect, but also to understand, for example, differences between different genetic variants. So there are many reasons why understanding natural history based on the genetic variants is very, very important. So there are a lot of work happening. I give you here an example, for example, that we are doing now for alpha dystroglycanopathy. There are ongoing work doing retrospective reviews at our end and differentiating between children who present with early onset alpha dystroglycanopathies compared to those who present later on. And similar studies are also done elsewhere in the world, like in the US. But I would mostly like to focus on LAMA2 gene-related muscular dystrophies to give you an idea of what we have achieved in the last years. LAMA2 muscular dystrophy is one of the most common forms of muscular dystrophies and usually presents from very early on in life and has relevant burden of disease with not only central nervous system involvement, but also respiratory involvement, scoliosis, and feeding difficulties. And many children need feeding support and respiratory support throughout their life. So there's quite a lot of comorbidities in these children. So understanding natural history and developing therapies is very important. There has been a lot done with case report studies, retrospective cross-sectional studies, and longitudinal studies also from our center. But most importantly, there has now been a big push to prospective longitudinal studies to really understand fully the natural history and identify outcome measures. And there are now 5 natural histories ongoing for this condition. Why so much interest? Because we are probably closer to therapies and there are many more outcomes from research studies that possibly indicate that we might have a little bit closer to develop some clinical trials for this condition. So we're really push from a clinical endpoint as well to understand better this aspect. The studies we have done so far really helped us to understand the early natural history of this condition. What are the abilities of patients with LAMA2 muscular dystrophies? Do these children able to sit and stand? And if they do, how the abilities change over time? And these observational studies really helped us to give an idea about change over time. We learned about respiratory abilities of these children. We know that children early on are prone to chest infections. And we know that when they're a little bit older, from the age of 5, and we can monitor the respiratory function, their spirometry abilities show a reduction in their respiratory abilities. And indeed, many children need respiratory support from around the age of 10 years on average. We also know that children who might be milder physically with motor abilities being milder may still present respiratory involvement. And again, this helps us to monitor these children better. Looking at the genotypes of children, we know that some genetic changes that cause loss of function of the gene can cause complete lack of the protein merosin, which is the protein produced by the gene. And these children often have more severe clinical symptoms. However, the variability in clinical presentation does not only depend on the genetic type, the genotype, so the gene change, or the presence or absence of the protein. So there is probably an effect of the rest of our genome. So still, there's a lot of work ongoing to understand the proper correlation between gene, expression of protein, and the symptoms. And then the studies also helped us to understand which are the good outcome measures. There has been studies done to identify the right physiotherapy assessments and outcomes to understand change over time. And the group in the US has identified, for example, this physiotherapy score called MFM that is quite a good outcome. And other outcome measures and biomarker are being now also investigated, like measuring creatine kinase in young age, or for example, using MRI imaging to understand correlation with disease progression and duration. So all of this is ongoing. Finally, I would like to highlight the importance that I mentioned, that it's important that patients avail good standards of care. And a lot has been done in general for congenital muscular dystrophies, but really what we need is a standard of care that is specific for LAMA2 gene-related muscular dystrophies. and similarly for all the other CMDs. So we started with LAMA2 again because of the need to have this urgently in view of the possible new clinical trials that we hope that will be soon developed. And together with the colleagues, together with Dr. Foley, Andrea Klein, and Alberto Zambon, we have arranged a meeting recently, really with the idea to start developing disease-specific standards of care for LAMA2 gene-related muscular dystrophy, and agree also on what are the best way to develop similar standards of care for other type of muscular dystrophies. And this work has been very useful, and we are now developing a guidance based on this meeting on different aspects of care, of MDT care for patients with LAMA2. And we hope that and the idea is that we will develop further meetings similar for other muscular dystrophies, and we aim to share all this information with patients and patients' organizations. Clearly, we also need to understand how many patients we have, and we need information from patients about also their condition and their clinical status. And apart from these natural history studies, I mentioned registries that are really important. And Sam McDonald from the Global 6 COL6 RD registry has given me this slide just to highlight to all of you the presence of registries available. One is run by CureCMD, which is the international registry for all congenital muscular dystrophies, but similarly, there is a registry for COL6 related muscular dystrophies. And these are some of the QR codes that you can use to log into these registries. So in summary, knowledge is important. Knowledge is always the first starting point to help patients, but also to help developing trials. It's really, we need to optimize care because we want to find cures. So really, the efforts that we're doing with natural histories and developing standards of care are relevant to improve trial readiness for all of these conditions. And implementing registries that are created by doctors, but also families and patients, is also a key step in this. We will continue to work at our end to identify new biomarkers and best outcome measures for each of these conditions, and work internationally to understand how best to develop this, because these are rare diseases. So really, we cannot work in silos. We need to work all together to develop clinical trials for all muscular dystrophies. Thank you.
Thanks, Anna. Brilliant talk, and thank you for putting up those slides about access to registries. It's possible people out there missed that. If they did, please write to us at either the information email address that was put up at the beginning, or you can write to us at research@musculardystrophyuk.org, and one of us will be happy to point you in the right direction for registries. I'm aware of the time, but there is one question from earlier on about what treatments and therapies are available, particularly for adults aged 30 and above for congenital muscular dystrophies. I wonder if you could very broadly speculate for a couple of minutes about what's out there currently, and maybe some themes for what's coming along. I'm aware that Severio covered a little bit of that.
So you mean the treatments for adults, that question?
Yeah.
Yes. So I mean, I work in a pediatric hospital, but clearly we transition patients to adult services as standards of care would cover also care for adults. So that is very important, that clearly adult physician also should, are aware of the standards of care. The type of therapies and interventions that are available for children perhaps might be not always applicable. Clearly, there is a difference, but I'm not aware that there is distinction. So the standards of care that we are developing would be, for all the patients. I mean, I would not expect that there should be a difference. So I would assume that it would be, disease specific, depending on what the condition is. So if there is cardiac involvement, they should have go to the cardiologist, but it's symptomatic therapies, as there are currently no cures as such and therapies that are specific for the condition.
Fantastic, thank you. I am going to move us on now. Thank you so much, Anna, for such an interesting presentation. If We haven't answered everybody's questions or addressed people's comments. We do have people's email addresses, and we will try to get back to you after this webinar with responses to your questions here. So I'm going to hand back now to my colleague Romla, who is going to chair the rest of the session on living well.
Thanks, John. Okay, we'll now move on to discuss living with congenital muscular dystrophies, or CMD. For anyone who has joined after we started, my name is Romla, and my role at MDUK is Information Coordinator, so I usually lead on the updating of our health and practical resources. Before we go into the Q&A, I just want to say that through our Condition A to Z section, we've recently updated several web pages, including a general overview on congenital muscular dystrophies, as well as pages on specific types. And that includes information on LMA, on LAMA2 related CMD and LMNA related CMD, covering symptoms, causes, and management, which we previously did not have. And we also have an alert card for congenital muscular dystrophies, which was reviewed and updated last year by a team of neuromuscular specialists. So this card provides information that could be relevant for people with any type of CMD and is suitable both for children and adults. And you can order that for free via our alert card order form on the website or by contacting our helpline team. And we're also very grateful to some of our panelists who kindly supported the development of those resources in the last few months and last year. So I'd like to welcome our three panelists who'll be joining Professor Tedesco and Dr. Sarkozy today to help us discuss and answer any of your questions. We're joined today by Victoria Crook, Dr. Elaine Chan, and Lindsay Pallant. Unfortunately, Emily, who is a speech and language therapist at Great Ormond Street Hospital, was unable to join us this afternoon. So if any questions come in about speech and swallowing, we're happy to pass those on to after the session. I'll now invite each speaker to make a few remarks about their experience of congenital muscular dystrophies and to raise some key points about managing or living with the condition. As I mentioned at the beginning of the seminar, we'll be covering as many questions as we can that have been submitted in advance or that come through the Q&A function, which you can find at the bottom of your screen. And another thing to note, our panelists this afternoon are specialists in paediatric care, so the discussion is likely to focus on the needs of children and young people living with CMD. Please do share any questions and we'll do our best to address them in conversation. And mentioned if there's anything we don't get to today, we'll follow up afterwards and get the information on our website if needed. So I'll now invite each panelist to introduce themselves, share their experience with CMD, and highlight any important points about living with and managing the condition. So is it okay if I go over to you, Lindsay, first?
Thank you very much. Yes, thank you very much for inviting me to participate today. And it's been really interesting hearing the research that's going on at such a very complicated scientific level. But I think it offers hope for the future. So as we said, my name is Lindsay Pallant. I'm a specialist physiotherapist based at Leeds Teaching Hospitals, which we're a regional centre for neuromuscular service in the north. And I've been working in this centre for 25 years now. And so during that time, we've obviously have a range of patients with the different extra rare CMD disorders. And so I've had experience of numbers are smaller, even in a regional clinic, which is a medium size in the north. Our numbers are small, but we've seen quite a number of children over the years, and I've had the privilege of seeing them grow. And that's the advantage of staying in a job in one place for quite a long time, is that you see the children when they're younger and possibly diagnosed very young, as we've heard already, but then you get the chance to see them grow and how things change for them and the impact that has on their life. And I think the thing that I've learned really as a physio is that my role really I consider to be thinking about mobility and function, and so that the children and the young people can do the normal things in life, develop, grow, go to school, and have the equipment and orthotics devices maybe to enable them to function to the highest level that's possible for them at the different stages of growth. And clearly there are stages where their bodies are changing a lot, from a baby to an under 5-year-old, and then as they grow through that primary stage, and then again when maybe get to puberty, the growth spurt again, how that can really impact function, joint range, mobility. So really concentrating from my perspective on mobility, function and activities of the daily living, exercise and activity, doing what you can, moving a little bit, posture and spine, really important, maintaining joint range to again optimize function. So those are the areas probably from a physio perspective that I'd concentrate on, but really for me the emphasis is on participation, enabling children and young people to be involved in their family life, in their community, schools, and with their peers, whatever that looks like at those different stages.
Thank you. Thank you so much for the introduction, Lindsay, and beginning to kind of pick up on some topics that we'll be covering in the questions as well.
Yeah.
Is it okay if I go over to you, Victoria?
Sure. My name is Victoria Crook. I'm a clinical nurse specialist at Great Ormond Street Hospital. And there's a team of us nurses that work together covering all of our neuromuscular population as such. So as we're children's specialists, we do a lot of support with schools and EHCP plans for families and helping communities understand the special needs and requirements in areas that people don't always think of in schools and things like that. So we spend a lot of time working with families to help them get some of the information across and explaining things, because it's a bit tiresome for them to have to explain the same thing every year in school, that they're going through the same things every time because staff have turned over and things like that. We also do a lot of support for the families with support letters for DLA, and the big one when they turn 16 and it changes to PIP, we also can help a bit with that. We provide a lot of letters for those sort of things, and day-to-day things, traveling, if people have to bring medicines with them, if people need special equipment with them, little things like that, that oftentimes families come to us with little problems that they've had, and then we get through it and we're able to pass that information on to the next family that might have a bit of an issue with something. So we tend to be the one-stop shop for lots of different problems, and if we can't find the answer, we'll often, refer out to the specialties, to the physio, to the occupational, to our respiratory team, to, whichever teams. We just sort of help to coordinate and work everybody together to be able to make things a bit easier for everyone to navigate, because obviously it affects lots of different aspects, and sort of everyone's a bit like, oh, who do I contact about this, or who's the best person for that sort of thing. So we find that quite a bit of our support as the nursing team is to help direct people in the best places to go for some help with whatever their challenges are at the moment.
Thank you. Yeah, I completely understand that there's so many other aspects aside from the medical care as well that are super important to families and children as well as adults too. Would it be okay if we come over to you, Elaine, now for an introduction?
Hi, my name is Elaine Chan. I'm a respiratory pediatrician working at Great Ormond Street Hospital since 2014. Before that, I was working at Royal Manchester Children's Hospital. So my experience kind of spanned from the north of England to the capital, and from the very beginning, since I became consultant, I have been involved in long-term ventilation, and my areas of interest include sleep medicine and long-term ventilation, as well as the general respiratory medicine. So it's very linked with all the aspects, the respiratory care in neuromuscular population. And in my current job, we are very fortunate that we have got a very high functioning sleep lab, which churns out over 2,000 sleep studies a year. And our cohort of patients on non-invasive ventilations comes up to about 350. So that is probably one of the largest single center cohort in the UK. And a neuromuscular section of the whole entire cohort makes up about 20 to 25%. So we are exposed to this group of patients very frequently. And I would say that probably over half of them have got CMD. So we're very blessed that we educated and experienced in this group of patients with the help of our neuromuscular colleagues. And over the last 10, 20 years, I have definitely witnessed changes, gradual shifts in the approach in the respiratory management. We blessed with the increased knowledge about the natural history that Anna was talking about. We are able to shift our approach from very reactive, when I first started being a consultant, to a much more proactive and anticipatory approach. And I think that is probably one of the biggest shift I see in my profession, where we are able to monitor and do surveillance of our patients and preempting when declines is happening, and hopefully helping our patients in a timely manner rather than when they have crumbled. And so, I mean, my work would be involved in the management of respiratory complications, probably the biggest cause of morbidity or mortality in this group of patients. So managing recurrent chest infection, swallowing dysfunction, aspiration, and then the sleep disorder breathing, supporting them with the non-invasive ventilation, and some of them may be invasive ventilation. And more recently, in the last 10 years, we are putting these patients through surgery under greater and greater skill and experience. So we are also involved in that kind of setting in risk assessing our patients, making sure they get through the surgery in a safe manner. So I would say that the biggest theme in this management of this group of patients is multidisciplinary working, and we benefit from having our neuromuscular colleagues to educate us about diseases and their natural history. We also get the help of a team of nurse specialists in long-term ventilation and respiratory physiotherapists who supported a big part of the management, and of course my sleep team, who help us to interpret, you know, when we should intervene and how the breathing changes during the sleep, when we should be intervening to prevent further deterioration. Thank you.
Thank you so much for explaining that for us, and thank you to everyone else for the introductions as well. So just a reminder, you can still submit questions in the Q&A function, which we'll raise. But otherwise, I'll begin with the first question, which might be relevant for you, Elaine, but anybody is welcome to please chip in with any answers. But we've got one question that says, if our child gets a cold or a chest infection, what should we do differently?
Well, I think common sense is still the most important thing, and it depends on the severity of the weakness and the previous history in the child as well. But I think, you know, observation, making sure that that you don't let the child get into kind of increased work of breathing without support, and being proactive in seeking medical assessment for chest infection and treatment, and assessment for chest infection is very important. And if the child is already on non-invasive ventilation, already on respiratory support, then usually the tertiary center would provide a escalation plan on what to do when patient deteriorate, making sure that the secretion management is on, you're on top of secretion management, and making sure the patient is well hydrated and the nutrition support is not neglected, and also having a decided or pre-planned threshold to seek medical help in the hospital setting, or phoning up the team, the medical team in the tertiary centers to ask for advice, and updating them on the progress of the child is really important.
Thank you so much. I have another question here that is about joint stiffness. So I appreciate that we are covering a group of conditions, so the severity can vary. But generally speaking, we have a question here that says, what exercises, stretches, or equipment could help delay joint stiffness and keep my child as mobile as possible? And who would be the right person to help with this? I'll open that to anyone. Lindsay, would you like to go ahead?
I'm happy to start, thank you. I think obviously the diseases vary, and I think there is certainly with younger children particularly, we would always start by promoting function and activity. So so let's just take the arms, for example. Though, if for a child who's able to move their arms a little, you would encourage activity through play, and therefore you'd be promoting joint range, you'd be promoting muscle extensibility and flexibility through function. And that's probably always the first port of call that as we use our muscles for function ourselves, to reach things, to play with toys, for standing up, then that's the best way to stretch, to maintain muscle extensibility, and it's the best way to maintain joint range. But for children whose limbs are maybe very weak and maybe don't have anti-gravity power, then assisting a movement is the best way to do that. So ideally, we wouldn't necessarily be looking to do stretches to a child or young person, but we'd be asking them to join in. Now, clearly, if they're 2 or 1, they're not going to be able to join in quite the same way. But we would say that stretches would be active assisted, whereas the child's asked to help. So if you're bending their leg up, you'd say, right, can you help me bend your leg up? And now we're going to straighten it down. Because it is easy to overstretch joints and overstretch muscles, particularly when children are very weak, because they don't have that natural protection of joints and muscles because of the level of weakness. And so joints that can't be moved through full range actively, so if a child isn't able to lift their arm above their head actively, they could be assisted to do that by physically someone helping them do it. But to try and make it fun at the same time would be optimal. So I think active assisted movements, getting a child to join in a movement, that could be done by doing something like going swimming. So it doesn't have to be on land. If you can find a way of doing an activity where some of the weight is taken for the child or young person by, for example, by the water, then they're more likely to be able to move themselves because of the buoyancy. Again, it's a fun, normal activity. And as long as it's safe to do so, you can do stretching and keep joints mobile by doing exercise and activity that way. It could be in a bath. It could be in a paddling pool. It could be in a hot tub. All these things can be a useful way of maintaining joint range and mobility. I think the best person to advise would be a physio or an OT on stretching and joint range. We would only stretch muscles and joints that are tight. In some of these conditions, you can be hyperflexible and lax. So it's important only to stretch joints that are tighter. Some of the diseases, they can be quite fibrotic. So as we heard earlier, that we've got dystrophic muscles that's damaged, and it's replaced by sometimes fibrous tissue and fat. So sometimes it's actually really difficult to stretch muscles, particularly as young people get older and into adults. The muscles aren't as extensible as they were when maybe the child was younger. The joints can become stiffer. So obviously we're trying to minimize the stiffness as a young person grows. But we also have to recognize that there is this underlying disease process and there's this element of fibrosis. And I think it's okay it is important to recognize that we can't stop contracture formation. We can hopefully slow it down. We can try and maintain symmetry. We can use equipment to do that. So good postural support in a wheelchair. We can maybe use orthotics. But unfortunately, the underlying disease is such that we're not going to win, especially since the child's body's growing. And changing all the time. I said earlier, through puberty, you get massive bone growth. And if your muscles are already a little bit tight and you get this bone growth that is quite dramatic at times, then the muscles become even tighter. The joints can become stiffer more quickly. So I think it's about thinking about what am I trying to do at the different ages and stages. Prevention is better than cure. Can we keep things mobile? If they're mobile, can we keep them mobile? If they need help, use a bit of help. if the child needs help to move things, to try to keep that range there. It's very difficult to get joint range back when stiffness has developed. And it's also possible to overstretch. And I think stretching can be a burden for a family. So kind of trying to work it into function and activities and fun things is really important, because I think the burden of care can be huge, and with that comes a package of guilt that we haven't done things as well as we might have done if only. So I think it's complicated, but I do think specific advice for a child or young person from a physio, working with an OT maybe together, they're probably best placed. But there may be other colleagues that can advise on that as well.
I just wanted to also just echo, yeah, like you said, trying to make it as fun as possible especially for a child or a very young person. That is kind of the way possibly to make sure that these exercises and movements last for a long time, rather than it being a one-off, because ultimately if you find something fun, you're going to do it more often as a child or an adult. So I completely echo what you're saying there. Also, just could you clarify how somebody would access a physiotherapist or an OT if they don't currently have that?
Yeah, certainly. So if you've got a diagnosis of a congenital muscular dystrophy, you should be under a specialist centre. And to have a diagnosis, you probably have been at some point under a specialist centre, but everybody should have access to a physiotherapist. So that would usually be via a community physiotherapy service, but your regional centre would be able to refer you to the community physiotherapy service. You probably, if you're a child, you should be under a community paediatrician as well. And the therapy services are linked with the community paediatrician services. There are obviously private providers as well, but I would always recommend an NHS provider in the beginning, because I think we, as a regional service, we'd work very much alongside the community physiotherapy teams and they have the experience of a vast array of they're working with a vast number of children, and they would have the experience, and we can liaise with them. But there absolutely should, in paediatrics particularly, there should be access to a community physiotherapy and occupational therapy service in your community who would link with the regional centre.
Thank you so much. We do often get queries through our info helpline and inbox about how do people access these services that are so vital to managing these conditions. There is one question about contractures that I'll just come to now while we're on this topic. It is raised to adults, but I'll just go over it. So would contractures in adults stop, or will they continue? If you move quite a bit, will it stop, or should you be stretching more?
Well, in my experience is paediatrics mainly. What I have found through my experience is that moving is good for us. Generally we are made to move. I think the contractures can develop quickly through periods of growth, and obviously once you're an adult, you aren't great. Your body is not changing in the same way as in paediatrics. However, through periods of inactivity, through a change in maybe work pattern, the idea somebody sitting down a lot more and doing less walking, or maybe other general health things happen, and that lifestyle changes mean that we maybe don't walk as much as we did, or we find that we're able to walk less than we could. So I think our bodies can still change. I think it is very difficult I wouldn't say they never progress. That's what I wanted to say. I think they can still progress, but I think the period of rapid progression tends to happen more when you're growing. But I absolutely am not experienced in the kind of 30, 40-year-olds so I don't know if anybody else has got more advice on that and better experience. But I think it is related to activity and exercise, and also aging will affect adults. So there will be that will have an effect, and an element of deconditioning sometimes. So I think it's multifactorial.
Yeah, thank you. I do appreciate that. Oh, sorry, do I go ahead?
No, I just wanted to echo what Lindsay said, that I think that's correct. And indeed, usually, at least from some of the adult centers we work with, perhaps the physio provision in adult services is slightly different because of because of the fact that clearly in a grown-up person, perhaps the changes may be less rapid, but nevertheless, the larger neuromuscular centers from an adult neuromuscular centers usually do have specialist physiotherapists. So we would also recommend that adults, wherever possible, are seen in a specialist center so they can review these questions with their specialist physiotherapist, who may advise how frequently they need to be seen and so on. But yeah, it's what Lindsay already said is.
Thank you. That was really helpful. And of course, yeah, I do appreciate that we're all pediatric panelists today. So I'm happy to look into that further after this discussion. I have a question from an individual whose child is starting primary school soon, and they are quite concerned about using a care plan in school in case of sudden illnesses or hospitalization. So it would be great if we could perhaps speak about the use of care plans, and especially in an educational setting, if that's possible. And just to kind of talk about who would support this and just quite an introduction, if that's possible.
I guess it probably depends on what the care plan is covering, in which sense of who would be mostly responsible for helping with it. If it's sort of general and getting the nursery setting used to the child's needs, it very well could possibly be a nursing team such as, you know, us at Great Ormond Street, that kind of thing, depending on what specialist center they're under. But the you know, care plans are fantastic, and it provides a standardized, you know, set of care that should be delivered for the child. So we would always encourage families to get one established with schools. Sometimes it's difficult to get them to follow it, but if a school or nursery or educational setting is even willing to set one up, then usually we're in with the better chance of them being able to help support everybody with that child's needs. So I'm not sure if that's really answered.
Yeah, no, that's a good start. Thank you. I also just wanted to kind of pick up. So when there are these possible issues within EHCP in schools, who is available to support perhaps the parents?
It depends probably on what exactly the problem is, if it's something that we can help support with, or if it's more of a social need, as in, you know, it's more that it isn't their immediate health concern, but it's a general type of issue. So, but as the nursing team, we'd always be able to sort of signpost families as to, if it isn't our you know, if we're not the best person to do that, then we would be able to signpost the families to who in the community might be able to support them better than we can from the hospital setting.
Thanks. Yeah, and I think from a charity perspective as well, if somebody wanted to get in touch with our
Absolutely, charities
info
are,
inbox or helpline, we can either support or definitely signpost somewhere more local, like you just mentioned, depending on what the situation is.
Yeah.
I'm going to take one question that's from the Q&A chat, and that is on ventilation. So this individual has said that they have congenital muscular dystrophy and their CO2 levels are high and may need ventilation for the nighttime. And they are wondering if there's anything that could be done to help prevent needing ventilation.
Okay. So I think from a respiratory perspective, the thing that one can do is to try to preserve the lung health as much as possible. So kind of trying to prevent worsening of lung function by preventing or managing chest infection in a very proactive way, having low threshold of using antibiotics and prepared to be using it for a longer course for treatment of chest infection. And if there is recurrent chest infection being the problem, then consider prophylactic antibiotics as well. Obviously, the day-to-day management of secretion management with respiratory physiotherapy and controlling excessive secretions are also important. There are other conditions that can exacerbate recurrent chest exacerbations, like reflux, swallowing dysfunction causing aspiration, and these also need to be tackled, assessed, and managed. And one of the things that we do for our CMD patients is regular surveillance, especially if they've got symptoms and signs of sleep disorder breathing and declining lung function. So the sleep studies will inform us about the severity of not just the CO2, but also the level of sleep disorder breathing, and whether one should take action. I think if it reached the threshold that we think that the patient is symptomatic and really needs some help, then it's always good to kind of embrace it rather than resist it, because in the long run it will help you to have a better quality of life. The other thing that would also make someone as a stronger and more resilient is also having a good nutritional support. We find that malnourished patients, when they come off, surgery or anything, they often require support longer from a respiratory point of view. So I think being well nourished and able to fight off infection better and therefore preserving your lung health is also helpful in this scenario. There are other things, of course, that not just the respiratory people can contribute to helping somebody to delay and a delay in need for ventilation. And I will be looking to our colleagues in neuromuscular specialty when they have got emerging kind of life transforming therapies, and then they may preserve the muscle strength so much so that that we can delay needing or not needing at all respiratory support one day. But I think these are things that we are kind of looking very far ahead in the future rather than right now.
Thank you. And you mentioned respiratory therapy while you were speaking, or sometimes known as chest physiotherapy as well. Would you be able to maybe speak a little bit more on that?
Ooh,
or perhaps let us know who would be the one facilitating that sort of thing for your patients?
I don't really think I'm the expert in
No,
chest
okay, that's
clearance,
fine.
but I would say that there are different methods of chest clearance and secretion management. Of course, our most basic thing is kind of making sure that the secretions are not stagnated in the system, and you prevent clogging up the airway, and a combination of physio maneuver, and with or without other devices to assist you, like cough assist and so on, is something that is a very specialized area that our respiratory physio will be in a better place to comment on that. But there are medications that can control the excessive secretion, like glycopyrronium, hyoscine patches that we use from time to time, especially for patients who are not able to control or swallow their secretion. This may become handy, but I would just say a word of caution that one should not try to thicken secretion too much, especially during an acute respiratory infection, because you do want to get rid of the secretion rather than keeping it in the airway and in the lungs to cause further problems. So and in the longer run, if the saliva or secretions is difficult to control, there are other means to consider, like Botox and salivary duct ligation that's more invasive. So these are kind of gradation of approaches as the problems manifest itself in different level of severity.
Thank you so much. I have another question here, which is, so how do we know if my child is in pain or uncomfortable due to muscle tightness or weakness, especially if they find it hard to communicate this? Would anybody like to answer this one?
Maybe I can just comment that clearly when there are contractures and positions that perhaps can cause pain, it's a different matter, but muscle condition in itself, muscle weakness in itself usually would not by default associate with pain. There are some conditions where pain can be more common, muscle aches and pain, myalgia, and we do know that some of these conditions can more often cause myalgia. So it's something that the specific diagnosis can also help if you know what type of condition you have, you can have an idea if muscle aches and pains can be present. But it's not the weakness in itself, because not all condition where you have weakness would have pain, if that makes sense. And then, of course, when there are tightness and contractures and unusual and with postures and positions that are not typical, then those could cause perhaps pulling and aches and pains that could be therefore managed with physiotherapy. Maybe Lindsay, you can, I'll just let you comment on that. But this is something that I guess you often also understand also for families where a child or person could have communication difficulties, that they may recognize that the child is uncomfortable, and that may indicate that there is some discomfort underlying.
Thank you. Yes, I mean, I can just add there, I completely agree that they're not necessarily stretches. Any physiotherapy any activity, but any physiotherapy activity, particularly stretches, should not cause pain. Stretching should not be painful. Stretches should cause a pulling sensation, but we would not want to be putting children, young people through anything which is a painful procedure per se. They're not going to enjoy it, and they'll be of no benefit to it. So it's very important that physiotherapy doesn't cause pain. I mean, usually parents are really intuitive with their children. And usually you would know if if your child is unwell, or they're too hot, too cold, or hungry. I mean, sometimes it's difficult to guess, but hopefully you would know if your child was in some discomfort. But stretches should not cause discomfort. They may be pulling, but they should not cause pain. And we just need to be very careful, because we know that children, when a child, young person can't move their joint through a full range themselves, they can't move their hip, they're spending a lot of time sitting, so they're going to be stiffer at their hips and knees. We do need to be very careful when we start moving, and we go very gently. In the beginning, we do a small range movement if we're helping them to move their leg, and then we would widen the movement and get the child to join in as far as possible, so that that you're not doing something to a child all of a sudden. You're warming the joint and the muscle up. Just like if we think somebody doing something to me, I wouldn't want them to come and pick up my arm and just move it very quickly. Hopefully they would take care. They would start slowly, start small, make the movement a bit bigger. And I think if you're doing stretches on a regular basis with a younger child, the carer, the person doing the stretches, does get a sense of the movement of that joint, that how well that leg moves, how much it moves. And they get a sense when they get to the end feel. And the end feel, we would say, would still be soft. We're not really pulling to the very extremes of range. If you were stretching a knee, for example, we wouldn't be advised. We obviously want the stretch to pull, but not to the point where you're feeling it absolutely solid, because that's probably going to be uncomfortable. So stretches should still have a softness. They should feel a flexibility. Yes, you're going to the ends of range. You might hold it there, but you're then not applying a lot of force. So hopefully, and if they're done regularly, then the carer, the handler would know kind of they get the feel of the leg or the arm or whatever it is you're stretching, or the neck, for example, because that's another area where contractures can be quite problematic. Get kind of a feel of it. So I think there is something in the regularity of doing things and the skill of the handler, and that, parents are absolutely expert at that. And they'll say to you, oh no, we don't move it like that, we move it like this, because this is more comfortable. So finding the positions to do the stretches, and you can usually tell by a child's face. But obviously, if there's a facial myopathy or other things, you would have to go by noises or your intuition. And if you're not sure, then you just stop and ask advice. I think that's the thing to do. Don't feel you have to continue with something, even if you've been told by a physio. Just if it doesn't feel right, stop and say, I'm not comfortable. Can we go with this again? And I think that's really important.
Thank you so much. And there is one other question that's slightly related to this, and it would be whether there are any small signs that can be looked out for that would indicate worsening posture or scoliosis, possibly in children with CMD.
Lindsay,
Would
you
anybody like to do? Anna, do you want to take this first?
So sorry. So the question is, what are the signs of worsening posture and possible scoliosis?
Yeah,
right?
things
That's
to look
correct?
out
Yeah.
for possibly.
So we usually do monitor these aspects in clinical assessment. So children with CMDs, depending on the diagnosis, but generally would be seen every 6 to 12 months. And an assessment of the curvature of the spine would be part of the neuromuscular review by the doctor, but also by the physiotherapy team. We would examine the spine. I would look at the curve, and if appropriate, we would do an X-ray of the spine to look at the curvature and monitor this a bit more in detail. And if there is a curve, then this can be monitored regularly with more frequent X-rays. Again, this would be decided by the degree of the curve and by the condition and so on, and the age of the patient, because we don't want, we would anyway want to do no more X-rays than what is necessary. So regular assessment both in clinic and with investigations.
Thank you, Anna.
I could just add there that symmetry is quite a key thing for looking at the spine. So if a child is able to sit, or if they're in some supportive seating, if there's a symmetry in the level of the shoulders, in the level of the hips, are the legs sitting parallel on the seat or on the chair, and is there a symmetry at the pelvis, or is there a tendency to lean? So symmetry is actually quite a key thing that we look at, as Dr. Sarkozy just said. And if the child has got their own sitting balance, then you can, and even if they're leaning a little bit, but they're free sitting, then that's fine, because what you're actually looking for is flexibility. So it's symmetry and then flexibility in that curve. So even if a child does tend to lean a little bit, if that curve is still quite flexible and that actually they could go the other way, then that's actually not necessarily a bad thing. Obviously, ideally, their body would be held in midline with the ability to go one way and the other. But if they tend to go more one way than the other, but actually they can still come back to the midline or back over the other way, keeping a flexibility in a posture is as important as the symmetry. And again, these aren't things that we can necessarily prevent totally, but we can help with management, and that is through good postural support, encouraging mobility and flexibility where you've got it, different positions, so that somebody's not in one position throughout the day. So, but early changes would be slightly altered symmetry, and any stiffness in that symmetry would be something to watch out for.
Thank you so much. Yeah, we are updating our webpage on posture and also reintroducing a new one on scoliosis. So they should be two very helpful resources as well for people in general. Not with a CMD focus, but it will be with muscle wasting focus, of course. I'm going to take one more question just from the Q&A, and this is to do with adults, but we'll just try and go over it. And that may be the last question in case anything else pops up. So this individual said that they're 33 and they have an undiagnosed form of CMD, and they are now struggling to swallow sometimes. So whether they should speak to their GP or their consultant at their clinic. And the individual has also said that why does physio stop at adulthood, and how it's still very much needed. So perhaps if we could maybe begin with the question about swallowing.
Maybe just can comment. I mean, I definitely would have this checked out, and if they are under neuromuscular center, I would definitely contact their center and request a review, and perhaps the clinician might then decide if they need to refer to speech and language therapies for swallow assessment. Clearly, this is a bit more specialist assessment where knowledge of the diagnosis and the condition, even if genetically undiagnosed, would be relevant. But I would definitely follow this up. If the person in particular who is messaging is not under an adult neurologist, I would ask the GP to make a referral as well. Because perhaps the diagnosis was not possible some time ago, but maybe nowadays it's possible, and maybe that can also help with the management. But while in the meantime understanding the issue with the swallows, not to wait for that, of course.
Yeah, definitely to try and get an assessment, and if needed, a speech language therapist could be very helpful with learning how to manage that, even if it's just early kind of symptoms. And then also just to pick up the question about physio. So physiotherapy access, I appreciate it can vary quite a lot, and it could sometimes be in bunch sessions, so it might not be ongoing. So just wondering if anybody in the panel could perhaps advise a bit about accessing physiotherapy as an adult, and perhaps if you has any like tips or anything to say.
Well, thank you. I mean, I think it's right that physiotherapy, adult physiotherapy is quite variable. A lot of the regional centres will have an adult physio attached to their adult neurology unit, an adult neuromuscular physio. We have an adult neuromuscular physio in Leeds. So there is somebody with that specialism in our area. And I think that is replicated around the country, not everywhere. Now, they may not necessarily be able to offer a lot of ongoing physiotherapy sessions, but my colleague Emma is certainly able to offer one-off sessions or see a patient a couple of times, and then if necessary she could then refer into an adult neurological physiotherapy team for, yes, as you say, like a package of care. And it does tend to be a little bit needs-based, so somebody's had an injury or something's changed and they can get I'm not pretending it's brilliant, but I think, I mean, obviously, I don't know where this individual lives. It maybe is unfortunately very difficult in your area, but I think if you do pursue it, you could try and pursue it and see what is available through your regional centre, wherever that may be. There are people who are really interested, adult physios out there who are really interested in neuromuscular, and so there's good care in places, but I'm sorry that that's not been something that's available. I think things are changing in the community. Every time I get my journal, they're saying there's more of this. You know, I think services are moving out into the community nationally. It's going to be a while, but I think that there can be, and it will be worth searching to see what is available. And maybe you'll have to try that quite hard, but I do hope that if there's something that you really need, even just go back to your GP and say, is there anything that I can have? It's worth pushing.
Yeah. Definitely. So of course, if they're able to access a specialist neuromuscular physiotherapist, that would be amazing through their centre, but there are some occasions where that might not be possible.
Yeah,
so
absolutely.
If that is the case, do try and perhaps get a local physiotherapist who may be able to assist you for a few sessions and provide some useful tips perhaps. So that's what we'd recommend. But of course, yeah, the need for physiotherapy and exercise continues in adulthood, and it can sometimes become even harder to manage. So I really appreciate that comment, and we do completely agree that physiotherapy or hydrotherapy is a crucial thing that we do need to improve access of for our community. So I think that will be the end of the question section. Thank you so much, all of you, for staying on. So as we come to the end of today's session, we'd like to thank all our speakers for sharing their time and their expertise. And a big thank you to all the attendees for any questions, comments, and their engagement throughout the session. The recording of this seminar, along with the others in the series, are going to be available soon, both on our website and on our YouTube channel. And we'll also be sending out a very brief feedback form, which we'd really appreciate you taking the time to complete, as it helps us plan and improve these future events. And finally, just a reminder that while today's session provides valuable information, it does not replace the advice from your healthcare team. And if you have any specific concerns about or questions about your health or care, we do encourage you to speak with your neuromuscular specialist or clinical team if you have one, or your GP. So thank you again, everyone, for joining us today. I really hope that you found it useful and informative, and I look forward to seeing you on the next session. Thank you so much, and thank you to all my panelists. Thank you.
Thank you.
Thank you very much.
Thanks.